This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 981, recorded on February 3rd, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here at the incubator in New York City, Dixon Despommier.
Hello there, Vincent, and everybody else.
You're right next to me.
I would love to tell you what the weather is outside right now, but I can't because the windows are shielded.
You do have a phone, right?
It's about, I would say about 12 degrees Fahrenheit.
Yeah, -7°C and very breezy.
And the chill factor is like down and below minus.
Very cold.
Monday's
supposed to be much
even
warmer.
Worse.
No, warmer.
Oh, no, it's Sunday and Saturday.
Sunday's worse, yes. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 13 Fahrenheit. So it's mostly cloudy, but it's really mostly sunny. And 13 Fahrenheit is -11 Celsius. Feels like -18 Celsius.
Right.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and it is exactly the same temperature here, 13 Fahrenheit, -11 Celsius, at least as of the last report, which looks like it was 15 minutes ago. It has been falling all day, and it's blasting out of the northwest at 20 to 30 miles an hour, the wind. So we have a wind chill of -16 Fahrenheit. So -27 C.
Vincent, if no one has heard
-21 C, sorry.
If no one has heard this before, which I doubt, but a lot of people out there probably haven't heard this before. Why don't you ask me how cold it is out there?
Don't say anything inappropriate, okay?
I won't.
How cold is it outside, Dixon?
Vincent, it is so cold that all the politicians have their hands in their own pockets.
Good. That was safe. Family safe. Very good. It's a family safe show.
It's a family show.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. It's 52 Fahrenheit, which I will tell you today is 11 Celsius. We are we are emerging from an ice storm. Storm kind of in quotes. That's, storm Texas style. It's not like, I lived 12 years in Buffalo. This is not right? But it did ice up. We had a really pretty morning on Wednesday morning with all sorts of ice. Locally around my place, we escaped damage. There were some trees down and some power out around, schools closed, all that kind of stuff. But everything's all cool now, or warm. We're headed back up to low 70s by Monday.
Really?
It's all good.
Wish I hadn't said that.
Hey,
Sexist.
we pay for it in the summer.
We made that happen, actually.
A couple of announcements for you. Research assistant position with Amy Rosenfeld at CBER of FDA. It's a part of FDA. They do research. This research will be on enteroviruses, genetic, biochemical, cell-based studies, all BSL-2, unlike some of the work we're going to talk about today in a moment. And you can find the email over at the show notes, microbe.tv/TWiV, where you can find Amy's email and you can get in touch with her about this position. Kathy, any news about ASV?
We have a lot of abstracts submitted. It's
Good
a
deal.
record number.
Wow. Good.
Wow.
Good.
Yeah, that's really exciting. And now it's time for everybody to start thinking about registering. And you go to the website and you register for the meeting. Whether or not you submitted an abstract, you can come to the meeting. And it's going to be a really good venue. And early bird registration ends, I think, sometime in May. So you have a little bit of time, but you don't want to get caught having to pay the higher rate just because you procrastinated. So that's it.
All right.
Oh, and the meeting's in Athens, Georgia, June 24th to 28th. You can go to asv.org/ASV 2023, or just go to asv.org and look for the meeting link and go from there.
And finally, you should join our Microbe TV Discord server, a place where you can go and chat with other like-minded individuals who are interested in viruses, bacteria, parasites, immune responses, neuroscience, all of the topics covered by our podcasts. And I think we have.
Stump grinding.
Yeah, you could. There are a couple of channels
there are a couple of lobby channels for off topic. For our,
I was
distant
thinking of
digressions.
choosing the Wuvel again for my pick of the week. But I didn't have enough time to do the research to see whether you can still buy them. Because somebody at some point did try to get one and they were out of stock or something. But I still like my Wuvel a lot.
Oh, yeah, I'm still a big fan of my Wuvel.
Yeah.
Anyway, I think we have over 500 members so far.
It's over 500. I just saw it today.
It's good. And always good conversations. And so Alan is there. We have there are what you call roles for people, right? There are members, and then there are admins, and then there are moderators. And so there is a Microbe TV host role. So if you are a Microbe TV host and you join, you'll be there. And people will talk to you and ask questions and be excited that you're there. So I think Nels has joined. Check it out.
And you can check into it whenever. It's a bit like the old Twitter. It's just a reverse chronological feed. You can see whatever's going on right now, like popping into your local bar. But it's all TWiV or Twix.
Twix. It's all Twix. All the podcasts are represented. Each one has a little channel. And it's been set up by an admin, Mr. Mason, who has it so whenever there's a YouTube post, it automatically gets pulled over there. Right? So the link, and then people can start talking about it. And same thing for my Twitter, same thing for my Instagram, for my lectures. Everything gets pulled over automatically, and then people can start talking about it. So it's fun. It's a lot of fun.
You know what, Kathy? It looks like maybe you can't get a Wuvel anymore.
Yeah.
You can get a snow
eBay.
pusher.
You could get it on eBay.
Yeah.
You can get things that have wheels that push the snow around, but it doesn't look like the genuine article.
Yeah.
I'll be darned.
Yeah.
Wow.
So I have a collector's item now.
Wow.
Yeah. You know, that could be worth millions.
Yeah.
All right, for our snippet, I want to talk a little bit about the recent meeting of the NSABB, the National Science Advisory Board for Biosecurity, and the proposal to expand the oversight on work with viruses and bacteria and other things.
Work that some people don't like.
That some people don't like. And so I wanted to tell you a little bit about the meeting, go into a little history of this whole story here, which actually we've been talking about the NSABB since the avian influenza experiments in ferrets of Fouché and Kawaoka, where they tried to get it to be transmitted by respiratory droplets. And that really brought the NSABB to the forefront, I think. And so I
think really the germ of this whole thing, if you will, was one really badly worded sentence in Fouché's original draft of his paper.
Yeah, I think that might be it.
I think that triggered the whole
the NSABB
thing.
was around before that, right? I think
it was, yes. So NSABB existed as a kind of like a government-sponsored journal club, basically. It was a discussion group more than it was not intended in any sense to be regulatory. And who did we have on from there? Was it
Mike Imperiale?
Mike Imperiale,
yeah, right.
That's right. And they got pulled into this all of a sudden and told, figure out what to do about this. And it was
actually they got pulled in before gain of function in a way, though that did get tossed in their lap. First of all, I looked it up. It was established in 1962.
Wow.
Right.
I didn't know that.
And biosecurity in 1962 must have been had a very different definition than it has today. My first experience with the NSABB, I actually visited these guys. Because if you will recall, some years ago, and my dating now, I have I have pre and post retirement eras. Okay, so this is in the pre-retirement era, which means it's before 2015, which is actually the whole gain of function flap was before I retired as well. But this was a couple of years before that when the dual use research of concern problem first arose, and I think that arose with the anthrax stuff.
Yes.
That was right on the heels of 9/11,
Right.
which then would have been 2001. And at that point, one of the things that came up was the whole dual use thing. Should we be publishing recipes for terrorists to use to engineer organisms to use as bioterrorism, right? And what came out of that ultimately was guidelines mostly for publication and for actual research. But where I became briefly involved was as on the editorial board of actually it was Journal of Virology. They sent me this thing asking, as a sort of a test questionnaire, this would be our screen, our filter for papers to look for whether or not they're dual use. And it was this big long thing, and I went through it and I said, man, actually I wrote back and I said, if I have to do this, I'm going to resign from the editorial board, okay? Because I think this is crazy. I don't think it's really as crazy as I thought it was then, but they hauled me in and wanted to hear what I had to say. But now there's a process that's pretty straightforward, at least for Journal of Virology, and I think other journals as well, where when you're looking at a paper, you check a few boxes, which, or don't check them, which, if checked, raises flags. So I think that was all resolved reasonably well, I think, at this point. And then the gain of function thing happened.
So gain of function, let's define dual use. You've used that term a couple of times. Why don't you define dual use and dual use of concern, right?
So dual use is basically, correct me if I've got this wrong, but experiments that you want to do that will advance knowledge on a given organism, but also have the potential to be used for evil, if you like, to hurt people or hurt things.
And this is a term that's been borrowed from the nuclear proliferation, chemical proliferation world. Dual-use research, you could be purifying isotopes in order to research, nuclear power, the whole sorts of good reasons you might be doing this, or you could be building a bomb.
Right.
So there's dual-use research that is...
That could never happen, though, could it?
And I think that they have this qualifier, dual-use research of concern, okay? Because I think there's lots of stuff that could be potentially dual-use, but there's no real concern about it being a problem. So
as the great poet Ani DeFranco once said, every tool is a weapon if you hold it right.
Is that like everything to something, everything looks like a hammer? What was that?
Right.
Including
like
a pen.
One of my favorite lines.
Yeah, if you're a hammer, everything looks like a nail.
That's right.
Yes.
Got it.
So, the way I read it now, gain of function, which we need to define as well, has become sort of a subheading under dual-use research of concern.
Yeah. So let me point out just some of the things that fall under this DIRC, dual-use research of concern. So there is a draft. We haven't gotten to the latest thing yet, but there's a document that's relevant to that. We'll link to. But in it, there's box one, dual DIRC policy scope, agents and toxins. So, for example, avian influenza virus. This falls under DIRC because if you're going to do something that has dual use, that's of concern because it's a highly pathogenic organism. Bacillus anthracis, botulinum toxin, Ebola virus, Marburg virus, 1918 influenza virus. You got smallpox on there, Yersinia pestis. So there's certain organisms on the list that would trigger a DIRC review. So if you're doing work on one of these, right, they're going to ask you, and at Columbia, you have something on your biosecurity, your biosafety form. Does this involve DIRC? And then every point elsewhere. If you send it to NIH, it's going to have that, and then if it's looked at by NSABB, right?
And work is also defined.
That's right.
There are 7 categories. So under the definition passed by regulators, DIRC includes studies that change a pathogen in ways that enhance its harmfulness, 1, disrupt immunity against it without justification, confer resistance to drugs or detection, increase stability or transmissibility, alter the species or type of cell infected, make the host more susceptible, generate an extinct agent on the regulated list.
Is Mastodon on that list?
No
Is
mastodon.
Dodo
It's
on that
not on
list?
the regulated list, no.
Mastodons are not on the regulated list. That's right. We'll get to that.
So, I mean, some of these manipulations are often done for agents outside of that list that I just read, right? So making organisms resistant to antibodies. We did that years ago for poliovirus. Nobody cared. We have vaccines. So I don't know about increase, if you want to increase the transmissibility, that was the avian influenza virus experiments in ferrets. So that would fall under this regulation because the agent is on the list and the experiment is on the list. Okay. So these were all put in place some time ago. I think right after the H5N1 experiments, if I'm not wrong, or maybe before that. But certainly the H5N1 experiments triggered more examination. And in fact, they triggered a moratorium on work with coronaviruses and influenza viruses and in modifying their transmissibility and so forth, which eventually was lifted some years later. So periodically, I think it's the NIH or Health and Human Services. Yes, Health and Human Services reaches out to NSABB with requests. And so in January of 2020, for example, they said, we want the process to be more transparent. The process by which all of this happens, where an investigator submits something and then it's checked. We want it to be transparent so everyone can understand what's going on. And so they had a meeting in 2020, and I went to that meeting, actually, which was highly contentious at some points, unfortunately. And
not yet contagious.
not yet contagious. And so
Beginning of 2020.
we
Pain of function.
nothing happened because of COVID then. I mean, we're all talking about COVID, of course. It came up multiple times. But because of it, that whole transparency thing fell by the wayside.
And I just want to point out at this point that even at that point in the discussion, the terms and the regulations already had a lot of vagueness to them.
Yeah.
So 2 things to highlight that popped into my mind were, first of all, increased transmissibility. What do you mean?
Yeah.
In what? Under what circumstances? So the original experiments that set this off were looking at aerosol transmissibility of H5N1 between ferrets. And what does that mean for transmissibility between birds or humans? Open question. And then things like increasing the host range. Well, this is something you would do routinely if you suit a type of virus. I mean, if you're trying to get it to grow and you put it in a different cell type, have you increased its host range? So there's a tremendous breadth of research, legitimate research, that falls under this potentially. And I think the way people have handled it since that time has been to just kind of say, look, unless it's really obviously something that's going to grab the headlines, it doesn't count.
Well,
that's my interpretation of the way this has been enforced.
Initially, it was restricted to certain pathogens, right?
Yes.
And that's where we are now. They want to expand it. But I also want to say 2 things. First, all of this is really driven by like 5 people who are worried about this sort of thing. And for some reason, they're very vocal, and everyone else is less vocal, okay? Secondly, I talked to a reporter the other day, and he was totally unaware of this. When they got avian H5N1 to pass from ferret to ferret by airborne routes, it lost its virulence for ferrets. Nobody remembers that. Nobody talks about it. He said, really? I didn't know that.
Loss of function.
Yes.
Yeah.
So it's like when you tinker with a lovely organism made by nature, you're going to mess things up, and that's never taken into consideration. So the disingenuous part of this, at that 2020 meeting, I was asked to give a summary, and so I complained about this, about how nobody paid attention to the fact that it lost its virulence. And one person who I will not name because people don't want to be name calling said, but Vincent, it was only 10 ferrets. And I said, when it was 10 ferrets that died, you worried about it. But when they didn't die, you didn't. I mean, it's so disingenuous, right? It fits your agenda, so you change it.
Vincent, can I ask you whether or not those 5 people that were so contentious, were they lab scientists or were they ethicists?
No, they're lab scientists.
They are.
I don't think any of them do virology, actually.
That's what I meant to ask.
Yeah, they're not. These are not. Generally, they're not people who are working in the lab with viruses on a day-to-day
That's what I wanted
basis.
to bring out.
So it's not a problem to be concerned. Okay, that's fine. But
Sure.
you have the same fine people now for 20 years have been pushing this and pushing it further and further. And I think they're just stealing the agenda from everyone else because the other scientists are quiet about it.
So has any examples emerged which gives them fuel?
So the reporter asked me that. Is there any? He said, I read all these examples of organisms that have escaped the laboratory.
Yeah, right.
I said, really? Tell me about them. The only one I know of is the
Smallpox.
1977 influenza virus that caused a pandemic. Is probably a result of a vaccine trial. Okay, so it was intentionally brought out of the laboratory. But, he said, what about the smallpox in the UK? I said, well, they were working in bloody laminar flow hoods. What do you expect?
Yeah.
Right, Rich? I mean, it didn't really escape.
Yeah, that was coming out of an era when we didn't have nearly the sort of biosafety that we have now, when people, at a time when there were labs that worked with smallpox out on the bench because everybody was vaccinated and it wasn't a big deal. Now, I don't think these labs were that insecure, but nevertheless, it was an era of much less biosafety.
Right.
As this a long time ago, lesson learned. It's not going to happen again.
So
And there have been a number of incidents over the years of accidental acquisition of a virus by a lab worker.
correct.
And these come up from time to time still. I'm not going to go example by example, but you can look this up, and it's something that has happened. It's something that continues to happen, and sometimes they're pretty dangerous viruses that get into somebody this way. But these are cases where it was somebody who was working with that virus and in some way failed to follow safety protocols or weren't provided with the proper equipment and whatever. This is a workplace safety issue. There's not, as far as I know, any recent, since this era of let's mouth pipette the smallpox, there aren't any recent examples of real outbreaks in the community happening from lab releases.
Right. So that's the issue that I was going to talk about a little bit, because I was sort of a secondhand recipient of all of the epidemiology with regards to loss of fever, because it was obviously discovered in Nigeria, but the first human case brought to the United States was a nurse that was kept in an isolation room at Columbia Presbyterian Hospital. And one of the workers of Jordi Casals at Yale, who received some of her blood to see if they could transmit this to other animals, caught the infection by a mistaken stick of a needle and went home and died, but did not transmit that virus to anybody else. So I don't know any major event that occurred with a dangerous virus that resulted in an epidemic. But I do know this, that shortly thereafter, because they wanted to work on this virus at the Army Research Laboratories in Fort Detrick, they invented the P2 and P3 facility as a result. So I think at that point, people begin to say, hey, maybe there's some other things out there that we have to be careful of.
Yeah.
And we need a better safety screen in order to do it.
And we're going back to the 1970s, early 80s
That's
when this
60s,
stuff was.
that's right.
Well, 60s, right? But into the 70s and early 80s was when the equipment was really
That's right,
deployed
that's right.
on a wide basis, and the protocols were adopted, and people really started paying attention to this.
Indeed.
So
Yeah, I wanted to round out the smallpox thing by saying it was late 70s.
Yeah.
There was one case of somebody who was not a lab worker, remote to the lab, and some smallpox from the smallpox lab, smallpox virus from the smallpox lab got through a vent
Right.
and infected this person. But it was one case, it was controlled, and the only other casualty was the lab head who committed suicide.
Because the person who caught the smallpox died.
Yeah.
Right, and there was a significant outbreak of smallpox in the Soviet Union around Sverdlovsk, right?
This one I'm not familiar with. This comes from one of these books.
No, this was
Conspiracy theory
this was a
book, did you say?
fairly well known 1970s incident where somebody changing a filter forgot to put the new one in, and it was at a Soviet bioweapons facility where they were growing up fermenters full of smallpox, and they sent some up the stack and had an outbreak in the city, which was very poorly documented because, of course, all the records were classified and then shredded. But
So both of those are basically failure of engineering controls.
yes,
Correct.
So the one case that Rich described and what you described, failure to put the filter back in, engineering controls are known. Failure of engineering controls can usually be traced back to some kind of human error, but you could
and
never,
in the case of the Soviet incident, that was something that was not even dual-use research. That was a weapons facility.
Yeah.
Right.
So there's not even really any ambiguity around that
Yeah,
particular
but there was a
case.
drug company in Germany that had an outbreak of Marburg, and that was
Oh, yes.
Well, they brought
another
monkeys.
incident.
Yeah, they brought monkeys who were infected.
exactly right.
Yeah.
So that was a veterinary problem.
But it was in the facility, right?
It was.
Right.
That's correct.
I mean, the point of all this is that the people who say we need to regulate more and more, they always cite long lists of accidents, but none of them really amount to. They're mostly contained.
They're
Right.
always thinking about what could happen rather
Yes, of
than
course.
what did happen.
I think it's fine to do that, but you have to also tell people that we have a wonderful biosecurity system in place with BSL-3 and 4 laboratories that are highly regulated, and they work really well.
Right.
And so if you want to do some of this research,
Right.
That's the way to do it, not to say you can't do it at all.
And I think the answer is also that when you're a graduate student, you get trained in this really rigorously, even before you learn
Sure.
how to handle, dangerous pathogens.
Yes.
And although the potential for these spreading infections is real, name one that actually occurred like that, and the
Right.
answer is
which,
none.
Which doesn't mean that it couldn't happen.
No,
right?
no. But you
the
could
point
say,
is, and people will always make mistakes, right? But you have to have redundancy built in. I mean, when we went to the needle, Rich and Alan, every system is like triply redundant.
2 + 1.
It's
Yep.
amazing.
They should be able to have two of the systems should be able to fail, and there's still one that's able to handle the full capacity.
I think the chances of this happening are less than the chances of the Earth being hit by another Earth-destroying asteroid.
Now, I think if you are working with known human pathogens like Ebola virus and Lassa, and you propose certain things, yeah, they should be reviewed and say, is this being done under the right containment? And that's great. But the latest thing now is to make it all pathogens.
Right.
Right.
And not just humans, but animals and plants.
Right.
And what is the impetus? Because nothing has really happened to spur this on, unless there's a science article where the author suggests it's because of debate about the origin of SARS-CoV-2, which is horrible if that's what's really spurring this on.
Yeah.
So, Vincent, you serve on the Biosafety Committee at Columbia. Is that
We
correct?
do. Yes, we do.
So, what sort of currently, if you're an investigator at Columbia, what sort of process is there that covers both dual use and gain of function, and how is that executed?
So if you want to do any experiments with microorganisms, you have to fill out a biosafety application, and on it, there's a place where it says, is there any aspect of this that's dual use, or gain of function, right? And if you do, you have to describe it. And then when it goes to the committee, we look at it and we discuss it. And it's very rare. So every time we have a meeting, which is monthly, the person will say, okay, next on the list, dual use research. Any dual use research of concern this month? No. Okay. Let's move on. That's it.
So
And then
let's say one does come up.
Right.
Am I correct that you would either try and deal with it locally or kick it upstairs to the NIH?
No, we would do it locally because they're applying to do the experiments, right? So, for example, one I remember was with diphtheria toxin, right, which fell on this list, and they adequately explained why it wasn't going to be an issue, and so we talk about it. But if there is a problem, we have to deal with it locally. Right. NIH, if they want to apply for a grant to do it, then yes, NIH will have to be involved as well. But,
and they'll have to fill out on the grant application.
Yes,
you have to go through a
that's right.
similar
The study
process.
section
right?
will look at it, and then it gets bumped up higher there, and eventually it could reach the NSABB, who might look at it, right? So
Yeah. So when I was at Columbia, also applying through ICAC to get my grant approved for trichinella work.
And ICAC is the animal IACUC you're talking about. ICAC is a meeting.
I didn't mean it. I meant that.
Ipecac is
So
for
I
digestive
got a
problems.
question from the board of directors for that group, and they invited me to the table, and I came, and they said, well, you're going to work on this in animals, right? I said, yes. They said, which animals? I said, mice mostly, but sometimes rats and rabbits. How is this transmitted? Well, I actually give this by lavage orally to the animals, and then they get infected. Oh, really? And then how can they infect each other? And I said, well, they'd have to eat each other in order for that to happen. And they said, oh, okay. And then it passed muster, but they'd never had this organism worked on before. So they had to go through the life cycle in order to know that it's a carnivore to carnivore, scavenger
No, it's
to
good.
scavenger.
It's good that they
And
brought
it's
you
not
in,
bad.
right?
It's not bad.
But
they
they
were doing
didn't
their
ask
job.
you if you. They didn't ask you if you would be eating the rats.
Well, I knew someone once that actually did that. I knew a
So
person who
anyway,
used to take the animals home and eat them.
So the latest is there was a meeting just last week where, the HHS told the committee that we think that there's some issues with this. We want to review. So they established two working groups from the NSABB who came back with some proposals. All right, and there's this draft report of these two committees which you can download and look at. There's also a summary in Nature that I'll link to. There's also a summary in Science, but I ran out of my 3 times looking at it, so I couldn't look at it anymore. So the one in Nature is, it's stricter U.S. guidelines for gain of function research are on the way. Maybe. Because
Maybe.
Basically the panel couldn't decide on it. But here are some of the ideas. First of all, the current definitions of a pandemic pathogen with pandemic potential or enhanced pandemic are too narrow. And they would like to extend all of this oversight for research on animal and plant pathogens as well as all human pathogens, not just those on the list. All right. There are many, many findings that I'm not going to go through, but one of them I think is very interesting. A determination of whether research meets the definition of dual use of concern will often entail uncertainty. It's hard to know because you're often not doing an experiment to enhance the virulence of something, right? But maybe it happens, and how would you know if it might even be a possibility, right? So I think, and the panel reflects that in their deliberations, right?
But you could do laboratory-based epidemiological work. I know that someone at Rockefeller was doing that with salmonella and mice, and they actually worked on trying to select for resistant mice that way.
Yeah.
Not the organism, but the mouse themselves. But yeah, you could have people doing population biology in the lab. So you could see whether or not gain of function occurred.
Well, right. There are cases where you would see it, but there are also cases where you might be just working with a virus and I don't know, you've got some mechanistic study you're doing, and as a result of putting it in your animal model, you made it more pathogenic. And you didn't know that going into it, that was going to happen.
You did not know.
Well,
That's right.
No.
the classic experiment is to take a virus that doesn't grow in mice and passage it
Passage
until it does.
That's
Right.
right. That's
And
right.
people did that for years.
Yeah.
It's how polio was adapted to mice in the 30s, right? They
Right.
passaged it. And that's one of the experiments that would raise eyebrows now. I don't know if it would for
Potentially.
polio,
Yeah,
because,
yeah.
but they want to extend this oversight to all human pathogens, not just of concern, but all. So poliovirus presumably would be under there as well.
And interestingly
Right. So
enough,
what happened was, with this report, the NSABB was commissioned to develop this report. End of January 27th, I think, they held the meeting releasing it, saying, okay, here's our report, and solicited comment. And the comment from the research community was all over the map. No, this is not strict enough. Oh my God, this is too strict. And just,
that's right.
it's a complete mess. Nobody could agree on this. But in order to change the report, they would have to reconvene the NSABB and basically send them back to square one. So they said, okay, we are accepting this provisionally as a draft, and we will continue discussing it. So essentially nothing has changed or gotten done,
No, not yet.
except that there's been a discussion of just how vague and difficult all of this is going to be if you want to do the stuff that these 5 individuals are saying we need to do.
Yeah. And they basically approved it, but they did so they wouldn't have to reconvene, as Alan said.
Yeah.
But they're going to have to modify the proposal and the report, and it's not even clear when that's going to happen or how they're going to modify it and so forth.
And the risk here is not so much that you come up with a protocol that's too loose, because as we've already discussed, we have these safety protocols from all their regulations that are already in place, but that you'll come up with something that generates so much paperwork that you grind research to a halt in exactly the areas we need to be working on to be ready for the next pandemic. And I think we've all had a pretty graphic lesson in how bad that could be.
Yeah. So one of the parts of this Nature article that's interesting is they have this statement in the draft that all experiments that could be reasonably anticipated to make a pathogen more transmissible or dangerous have to be reviewed. And they're like, what does this mean, reasonably anticipated? They didn't define what that means. And so the committee, we need to have a definition of reasonably anticipated.
And so I'm an old school person here, but I mean, I grew up in an era where they were looking at a very few infectious, effective vaccines. And one of them was yellow fever. And yellow fever vaccine actually occurred because they kept passaging a virulent strain to get an avirulent strain.
Correct.
And that approach is valid even today. So if you start with something, how else would you know unless you did the experiment?
Well, and today they would say, well, it could make it more virulent, so we're not going to let you do this except under BSL-4.
That's the point.
So we'd never have that vaccine.
Or everybody would have a BSL-4 facility, and that's ridiculous.
Well, we saw many places don't even have a BSL-3, which limited SARS-CoV-2.
There wouldn't be the funding to begin to have
Yeah.
that much BSL-4 and BSL-3
No money.
lab
No money.
space.
So I think my view of this is expanding this is absurd. I think you could have the original limits to these really dangerous human pathogens, but expanding it, in my view, doesn't make any sense. The chair of this committee said, oh, it doesn't matter. There are going to be so few experiments that fall under this. So then why do we have to expand it then if there's so few experiments that actually fall? So I don't agree. My personal view is I hope this never sees the light of day because it's not needed. I mean, even Mark Dennison said, nah, I have serious concerns, but he had to vote for it because otherwise they would have to have another meeting, which apparently nobody wanted to have.
Nobody wants to have this meeting again.
So despite everybody's major concern, the most dangerous place is outside the laboratory, not inside.
Yes.
Just to make that clear. There are more unknowns that we could encounter out there than there are knowns that we always work with in here. And that's the way to put it.
Who put this in here? CCBNA?
Me.
I'd never heard
Oh.
that before. That's an interesting one.
Oh, well, that's quite local. That was one of my college roommates.
Chit chat, but no action.
Chit chat, but
CCBNA.
no action.
Interesting.
There's a military term, a bogsat, bunch of guys sitting around a table, which is unproductive meetings.
Yeah. Well, this is a tough one. I think that, these are things that we need to think about and discuss. Fine.
Sure.
Okay. And I think it's good when, you just can't go off willy-nilly and do anything.
But,
right?
but people don't.
You need to
people
stop.
don't, right?
They don't do that. They don't do that.
No, but no, generally people don't. But it doesn't hurt to have somebody stop and read a thing that tells you what DIRC is and what gain of function is, or maybe have a lecture on it in your graduate training or something like that,
Right.
Or a little. Something from your biosafety committee when you're taking on your job or getting your grant, and make you look at this stuff and spend a few minutes thinking about it and make sure you're not going off the rails here. Check the appropriate boxes. And in some rare cases, maybe something needs to be bumped up, in terms of biosafety. But one of the things that comes to my mind is this: confer resistance to drugs or detection. Okay? If you have some drug, say, that you found that was a natural compound that inhibits the growth of some pathogen, how do you find out if that works? How do you find out how it works? The way you find out how it works is to isolate a mutant that's resistant to it and then map the mutant and figure out what the gene is. Okay? So
Yeah, I was just going to say, I have the science article that Vincent didn't have. I have that up, and many virologists worry that this could delay work with relatively benign agents such as cold viruses, herpes, and viruses modified to treat cancer. The rules could also complicate basic studies for exactly what Rich just said. Deliberately creating viruses resistant to drugs is essential to developing antiviral treatments. And so this is,
So early in the pandemic, a faculty member at Columbia was selecting for SARS-CoV-2 resistant to remdesivir. All right. So this came to our committee, as it should. And many people, not virologists, were upset about this. They go, why would you do this? And it could get out. But you need to know if you can get resistance. And if you do, could you make a derivative where you get around that? And that's often so you have to do that. And so we approved the experiments because you get useful information that outweighs the risk of them. And it's done under BSL-3, so that should be...
But Vincent, they're already doing that experiment. It's called giving the patient the drug
of course.
and then seeing what
giving
happens.
the patient the drug
Come
does that
on,
experiment
kids,
every time.
Let's get real about this. So when you
Let's do it
when you train somebody in the lab, and I know you guys are strict on this, you start a student out at stuff that won't harm anybody, and you teach them the safety rules, and you can tell when they screwed up because you get a positive where you should have had a negative, or a negative when you should have had a positive. And that's how you train people to be careful. You work with stuff that's not harmful first, and then move into the more pathogenic stuff.
The paperwork burden is a concern as well.
Yes.
Yeah, it is.
During my career, I saw the paperwork and the regulatory stuff grow and grow. And we need to have checks on potential problems. So a certain amount of this you have to do. But at some point or another, you need to at least carefully think about whether or not this particular bit of paperwork or regulation really is productive, or whether it's just getting in the way. Lead, follow, or get out of the way, right?
Right.
I mean,
Yeah, I do applaud the idea of making this whole process more transparent.
Yes.
fine.
Because the essential thing that really should be driving the whole discussion is this is publicly funded science, and we need to make it clear to the public how it's being done and how it's being kept safe. And if we're not doing that, we need to do a better job of it. So the paperwork should be to that end, say, so when some newscaster says, well, how do we know it's safe to do this? Oh, well, we know because we have these regulations that do this, this, and this, and here's the paperwork showing that they have complied with that. Or in the case of this accident that occurred and somebody in the lab got sick, here's the paperwork, the lack of it, showing that they didn't comply with the regulations, and that's what happened. So we do need the accountability, we do need the regulation, but we need it to be clear and transparent.
All right.
I want to point out something that's, we talked about the gain of function experiments, and full disclosure, I was one of the authors of the Virology Under the Microscope, A Call for Rational Discourse. I was in the alphabetical set, so I had a few copy editing and minor edits to the manuscript. But table 2 in that is, I don't think we talked about this before, is examples of useful gain of function experiments. And they've gone through and documented a whole series of things like insect control, the COVID-19 vaccine, enhanced lithium batteries, faster computers, drought and salt resistance in plants, resistance to freezing in plants and animals. It just goes on and on and on. CAR T cells,
Better rocket motors that you could also use for missiles.
Right. So anyway, it just shows and it lists the microbe and it lists what the gain of function was and it gives the references. So it's a really nice compendium.
Good.
Yeah.
Excellent. That's good to know. Yeah. Because that's what reporters always ask you. Give me an example of beneficial gain of function.
There's 20 here.
And I know that they'll counter Rich with, but what about the Borg?
Let's do something fun.
Yes.
Let's do a fun paper.
I could go for a bite to eat. How about you?
Yeah, I'm hungry too.
PNAS paper. The consumption of viruses returns energy to food chains. This is often comes up. People always say, could anyone eat viruses? Because there's so many of them around. John DeLong, James Van Etten, Zaina Al-Amili, Irina Argokova, and David Dunnigan from the University of Nebraska-Lincoln and the Middle Technical University in Baghdad. So this is very interesting. All right, so let's talk a little bit about viruses. Viruses infect cells.
Yeah, let's talk a little bit about viruses.
Hey, let's have a show.
Yeah, well, you know what I meant. The viruses infect hosts, and in some cases they lyse them, and bacteria or eukaryotic cells, and they release nutrients. In the ocean, this is called the viral shunt, when all those viruses, and there are millions of viruses per teaspoon of ocean water, they are causing many, many infections and liberating particulate organic material, which would normally be consumed by predators, and they are short-circuiting this pathway. It's called the viral shunt because the viruses are causing those organisms to break up before they're eaten. And so this paper addresses whether viruses could be used as a food source. And, a lot of things out there swallow water, right? Water, soil.
And viruses are full of protein and nucleic acids and
Indeed.
lipids.
But as they say in the introduction, given the small mass of virus particles, the consumption of viruses is thought to be calorically unimportant, right? And so ciliates and flagellates may ingest lots of viruses. And is this an issue? Is this a thing that they're actually eating the viruses, which would be called virovory, right? Is that how you pronounce it? Virovory?
Virovory?
Virovory.
Oh,
Virovory?
Virovory.
Or virophagia?
Oh my gosh. I knew we would have a fun time pronouncing.
This is called a gain of weight program.
Yes, it's a gain of And I have to say, so this is an open access paper in PNAS. It's a brief report. And it's almost, I got a sense of nostalgia reading it because the experiments are straightforward. Like just nobody'd done this before and they did it and they got very clear results. And the whole thing is just briefly, here's what we did, here's what we see, and here's what it means.
Yeah,
it's like a paper from the 50s.
It's like BSL 0.
Yes.
right?
Yeah.
A couple
Yes.
of drops of pond water, basically, in a petri dish.
A lot of early ecologists used these organisms to study escaping from predation, and yeah, it's very interesting.
Right.
to
So
see.
they're working mostly with Halteria, which is a ciliate it's freshwater.
Freshwater ciliate.
Right.
Yes.
Yeah. So it's go to your local pond or mud puddle and take out an eyedropper full and put it under the microscope. You could probably find this thing.
That's true.
And paramecium, another freshwater.
And paramecium, which of
ciliate.
course everybody saw in high school biology.
So this is a very simple experiment. I love this. We created foraging arenas out of petri dish. We put little drops, 0.4 ml drops, with the Halteria and the paramecium, and then they either added washed virus or a filtrate where they remove the virus, which is very good, right? Not PBS, because maybe the virus
Yes.
left something behind. So, and then they incubate those at 22 degrees Celsius, and then they measure whether the ciliates have increased in number and the viruses. This is a chlorovirus that they're using here, which is a double-stranded DNA virus, freshwater viruses, and the algae are the natural hosts of these chlorella viruses. It's a virus that we've heard many, many times from Jim Van Etten at giant virus meetings, right?
Right. And this is a virus, importantly, that cannot infect Halteria or paramecium, as far as we know.
The coolest thing about chlorella is that they can do plaque assays on lawns of algae. So the lawns are green and the holes are
real lawns.
yeah, and the holes are white. He shows this all the time. It's such a beautiful plaque assay. So there are just two figures. Or maybe, two figures with multiple
Two
parts.
figures.
The first one they're looking at for both of these ciliates, the number of cells over time. They're looking for at 2 days, 0, 1, and 2 days after they establish these. What did they call them? Let me look it up. Foraging arenas.
Foraging arenas.
The arenas
yeah.
reminds me of a Roman thing, right? Arena. They look at
A foraging arena, by the way, is a drop of water
drop of water on
Yes.
the plate.
on a petri dish.
Yes.
We who are about to eat salute you.
And they count the number of cells, and then they do a plaque assay to count how many PFUs are present. And you can see that with Halteria, with both of them, the number of PFU goes down, which they interpret to mean is the viruses are being eaten by the ciliates, right? And they have a fluorescent micrograph here, which appears to show
Sure.
viruses inside of the ciliates, right?
Yep.
But only the Halteria grow. The paramecium do not grow. So
This is not a complete diet for paramecium.
despite eating these viruses, they don't seem to be growing.
I was impressed with the fact that they're only putting in 2 times 10 to the 7th PFU per mil.
It's not a lot.
Now I don't know what the particle to infectivity ratio is on these guys, so it could be 10 or 100 times more particles.
Or thousands, yeah.
Yeah, but it's not a huge number. It's not like they're swimming around in virus goo or anything like that. This is it's not it's not out of the realm of physiological possibility, I think.
Well, maybe they should have put more virus, and maybe the paramecium would have grown. Maybe it needs more to grow.
Yeah.
Right.
Maybe it's just
Or
hungry.
A different food source. I think.
I think the Halteria would agree that it needs more. It's just it's devouring these.
Well, that's probably their natural food. But paramecium is an omnivore in terms of
Right.
what it eats. It'll take and eat particulate. So what about the following? If you had E. coli, which was infected with a phage, a lytic phage that you could induce, and you could prevent the growth of E. coli. So you're only looking at lysed E. coli versus non-lysed E. coli. And then you put paramecium in there. It's either going to grow better or worse, depending on whether you lyse them open or not.
Well, it might eat the E. coli too.
It's
right?
going to eat the coli. That's their food.
Oh,
That is their food.
But, so what are you going to look for in the end? I don't understand.
Well, if you and you can also have an E. coli without the phage.
Okay.
So you have a sterile
So
culture.
you're going to look for more growth with the phage? Is that what?
Well,
you're...
that I don't know. I don't
Because
know.
the
I didn't do that
lysis
experiment.
the lysis is not going to inhibit the growth because the pieces of E. coli after phage they're still going to be eaten.
Right.
No,
I think what Dixon's getting right.
That was another.
Does the phage fortify the bacteria and make it more nutritious,
Exactly.
or
Yeah.
by co-opting
Exactly.
parts of the E. coli has the phage made it less nutritious?
That's correct. And you can do with and without the phage to see if there's a difference.
Right.
That's right.
But in the case of Halteria, phage is virus is a complete diet, right?
Yeah, because the experiment
growing
viruses.
the experiment with filtered virus, right, where there's no virus, they don't grow.
Right.
In these pictures, it's quite clear,
That's fascinating.
right?
So they eat the chlorella to get to the virus.
No, the chlorella virus.
I
is
mean,
what
in
they're-
real life.
They're just eating the virus.
Yeah.
So they.
Well, in real life, this is.
No, it's a
Go ahead.
it's a side effect. It's it's a byproduct, if you will, that they can eat the viruses as well as. Yeah. I'm not
Yes, right. They
explaining it
can
very
eat the
well.
virus as well as the host.
Right.
But they don't gain with the host; they gain with the virus.
So there's no algae here. There's just virus. It's purified virus.
Right. They didn't.
Right.
I don't know. I don't know what their growth curve looks like with the algae.
But that's what's missing in these experiments.
Well, you don't
Okay.
need to. I mean, if virus is just floating around with, you don't need to invoke that. The problem with this virus is very interesting. Is that it infects an algae which is within a paramecium, right? And so, we did this paper on TWiV some time ago. The paramecium needs to be eaten by a copepod, which then releases the virus so that it can infect more algae. It's
I
a
wonder if
complicated-
Halteria eats paramecium.
I mean,
don't
I guess this
think
virus
so.
and Halteria and paramecium are found together in nature,
They are.
right?
They are.
So this makes sense, in other words, right?
Yeah, yeah.
And I think
ecological
Halteria
sense,
and paramecium are of similar sizes, and
That's right.
yeah, I don't think Halteria eats paramecium.
There's another protozoan. It's a holotrick called Didinium, and it eats paramecium almost exclusively.
So they have also in this micrograph in Figure 1 some other ciliates, and they say they also take up viruses. So let me play devil's advocate. So I don't see the viruses being digested. I would really like to see, I don't know, label up some viruses and then look at
Where the protein goes.
I'd like to see some follow-up
some
on this. yes.
biochemistry. I mean, this is a
Yeah.
PNAS paper, right? I think
Yeah.
could use a little bit more. I mean, what if the virus is just triggering some pathway that causes growth, cell growth? They're not actually nutritional sources.
But if
They've
virus
got
is
to
the
have
only
some
source
nutritional
of
source in
carbon,
order to grow, though, right? I mean, where's the matter coming
you need a
from to
carbon
build
source.
the new
that's right.
organisms?
You need a carbon source.
Okay. But I just think that the assumption that it's the virus is a little bit indirect, right?
And a nucleic acid source. How many nuclei
But they
does
did-
Halteria have?
Well, the
don't know.
virus is the only thing. That's the only protein source they're giving, right?
Right.
Well, yeah, I guess. And the
think so.
filtrate, the filtrate doesn't
So-
do anything. Yeah.
Right, and the filtrate doesn't do anything. The pure virus that has nothing else in it seems to be sufficient for Halteria to grow.
Yeah, and their control is a filtered version of the same thing,
Yeah.
so no virus.
Then I think that's pretty clear. I would still like to see how it's being digested and like how long can you carry this out? Can it survive indefinitely on virus alone, or does it have some nutritional deficiency later on that kicks in?
This means that, I mean, if it's true, this really means that nature abhors a vacuum, because no one ever thought of viruses as an energy source before. And here it is being demonstrated, at least in laboratory situation, that it might be possible. So how many viral particles, Vincent, is there per liter of seawater?
Well, per liter of seawater, more than people on Earth. So it's a few million per milliliter.
So, you know, if you packed it down into a test tube, could you actually see the pellet?
So 1 million per milliliter. That's on the order of what they did here, right, in the ocean. So it's physiologically
Sure.
relevant.
So
yeah.
this may be a huge source of carbon.
It could be. It
For
could be.
filter
Yeah.
feeders.
I keep looking around because I have this thing that says viruses, most of which infect microorganisms, are the most abundant biological entities on the planet.
That's right.
So here we have
So-
something taking advantage of that.
Exactly.
That's
So
fabulous.
why wouldn't you eat that? Yeah.
That's
So
amazing.
this has implications for the food web, right,
yeah.
Dixon?
It's food
Yeah.
chain. This is a food chain.
Well, they say current models of aquatic food webs and ecosystems
Webs.
do not include the trophic link between viruses and their consumers.
That is true.
So in other words, so ciliates can consume enough virus to grow. These are, and then in turn consumed by zooplankton.
Correct.
So they're saying this virus energy can move
This is
all
a
the way up.
zooplankton. No, these are zooplankton.
Well, they say these ciliates are consumed by zooplankton.
But this is a zooplankton.
Well, maybe other zooplankton. I don't know. That's what
Larger
they say.
zooplankton,
They
like
wrote it here.
copepods. It's
Larger
like copepods.
zooplankton. I think they mean multicellular
Or,
zooplankton.
you
So
know,
anyway, this virus can go all the way up the food chain,
They can.
basically. Food web,
Yeah.
they call it. Why isn't it a food web?
If you
this
have
is not a
more
food
than
web
one
because
chain,
you've
it's
only
a
got
web.
3 things in there.
Right, no, Dixon, they're talking about the bigger picture,
Yeah.
Where these
Oh, no,
things
I
are
got
floating
you.
around
I
in the
know what you're saying.
pond, and they are being eaten by the
But
bigger
of course,
zooplankton,
of
and
course.
yeah, that's a food web.
Yeah.
I have a resource here. I don't know how authoritative, but reasonably, that says a milliliter of coastal water contain up to 10 million viruses. So that's 10 to the 7th.
Yeah, that's right.
You get further offshore or deeper, it can go down to 10 to the 5th, 100,000 viruses per mil at a depth of 4,000 meters.
So they say here, we estimate that the Halteria ate 10 to the 4th to 10 to the 6th viruses per day, suggesting that 10 to the 14th to 10 to the 16 virions could be consumed per day in a small pond. And then they go on to the viral shunt, the lysis of cells is thought to limit the movement of energy up food webs by short-circuiting the grazer microbe
Right.
interaction. But our results indicate that energy and nutrients from microbes may pass through viruses into grazers and move up the food chain through virovory.
I like it.
Virovory.
Makes good
right.
sense.
By robbery.
Virovory.
It was highway for robbery.
So what is it, Dixon, the trophic link? What
Yes.
does that mean?
It's the energy link, and then they start with it's all about energy, right? So the primary producers are plants,
Mhm.
and so viruses can't be part of that, but they can be a parasite of plants, as this is, right? So this is short-circuiting the primary producer level,
Yes.
but something is there to take advantage of that even. So all the filter feeders benefit from this no matter what. I love this.
Well, assuming it happens in nature, right?
Well, if it happens in a test tube of this kind of organism, one might assume that it's happening outside.
Yes, but
It probably happens at some rate in nature.
Exactly.
They're the most abundant biological entities
Yes.
on the
Exactly
planet.
right.
That's a lot of
Exactly
protein
right.
to leave on the
so
table.
could we do an
Mhm.
experiment to see what's going on in nature? How could we do
Oh
that?
no, you could label virus
You could
particles
label-
with a
yeah.
C14, and then just see what happens to the C14, just like Eugene Odom did on the Savannah River project.
So take some seawater and add labeled viruses?
Mhm.
Okay. Yeah, I think taking samples and showing it. I mean, I agree with you that it most likely happens.
And you can do that
But
with the
I would-
nucleic acids
I
as well.
think you need to show it at some point, right?
Well, that's an easy experiment. That's dead simple.
And
Yeah, you could do it. You could do it progressively too. You could take it a bucket of pond water and do the C14 labeling, and you'd be able to sample it frequently right
Exactly.
there in the bucket in a contained system
Exactly.
that is effectively a pond, and see who eats what,
Yep.
and then track it up through there.
And you could use mass spec to see the. You don't even need to use
Listen
label.
to
I
us.
guess you could do mass spec and look at the viral.
Oh yeah,
Who's going to be the
Components.
principal investigator on this?
No, I'm retired with science, but
This is
So what?
this is really
what
an
are
easy
these-
experiment.
What do these organisms eat as their normal diet?
Halteria?
particles.
I do not know that, but I know paramecium is a generalist. Anything small enough to fit down the
like E. coli.
definitely.
Or pieces of E. coli,
Or
right?
pieces
Doesn't
of-
Or
necessarily-
viruses.
That's right.
Can-
Pieces of
Yeah.
anything.
Or
Viruses.
They eat other microorganisms like
They do.
bacteria and
They do.
algae. Oh, there's a mouth. Food
That's-
enters here. There's an arrow.
That's why I mentioned Didinium, because it only eats paramecium. So how does it do
You
that?
know, it would. Something just occurred to me. it would be interesting to do, if you could, do an experiment to see if these organisms would migrate up a concentration gradient of viruses.
Yeah.
Okay.
Yeah.
So,
Oh,
or
yeah.
if they would gravitate towards these as evidence that they were actively seeking them out as a food source.
They can eat them, but do they like them?
Yeah.
For that, Rich, though, you'd need to have pure cultures of each of these these ciliates, and then you would add each one to the C14 mix, and you could see how fast it actually got inside.
I would like to do it with different viruses to see, if some
Yeah,
DNA
I
versus
would-
Give them the
RNA.
Pepsi challenge.
Look at
See
them,
what
look
they
at them.
like.
Yeah.
What tastes better, DNA or RNA?
Who's
Yeah,
retired?
well, I mean,
Tell me
you
who's
know,
retired here. None of us.
Why the paramecium, don't seem to be eating it, but maybe some other viruses they would like a lot better.
Yeah.
Well, they are eating it. The PFU goes down, but they're not growing, right?
But I think paramecia are more
Right.
Instead
they're
of an
not growing.
arena, we need a
tuned to
buffet.
particulates.
Right.
And the viruses wouldn't classify.
But
as
DNA
particulates.
versus RNA, membrane, no membrane, giant versus small.
Yeah.
All these things you could do.
I think the biology of these two ciliates is different.
Yeah, very interesting. Now, the question is
Euplodes is another one.
do you
that they
think
could
it
have
would
used
just
here,
be plankton that are eating viruses, not larger? So when a whale takes in thousands of gallons of water, don't viruses go in?
but sure they do.
Sure.
But how they go right
But
back
the
out again?
baleen is
They
too.
go
not-
back out again.
Yeah, the baleen is not fine enough. It's not a 0.2 micron filter. It's only filtering out down
No,
to
but-
the size
but-
of like krill.
It is a membrane of sorts, so it could stick to that. And
That's
then,
a good. That's a
It
good.
could.
The baleen is not a 0.2 micron filter.
No, it is not. No, it is not.
Dixon, you mentioned euploides, and euploides is one of the ones they used
Yeah.
in this figure.
Oh, okay. Okay, good.
Yeah.
So these are unicellular ciliates,
They are.
right?
They are, but multinucleate. A lot of them are multinucleate.
And I bet some multicellular ciliates would also eat viruses. Right?
Multicellular. Name a multicellular ciliate.
I,
There aren't
Yeah.
any.
it's out of my lane. Well, what's multicellular?
A
Cellular.
copepod would be a filter-feeding zooplankton organism like
Copepod could-
cyclops.
So a copepod could eat viruses too,
Well, they could eat the bacteria that contains the virus.
not the virus themselves.
think so.
They're too big.
It's too small.
The copepod.
I bet.
Well, copepods have these mouths with little cilia in the front, and they just sweep everything in, and
Yeah.
it
Well,
passes out again.
what's the size of a copepod compared to a paramecium? Then?
Well, you know what Daphne is, right? Because
Yeah.
they conducted early phagocytosis experiments with it. It's about that size.
So the
It's a
viruses
micro-
are entering.
It's a microcrustacean.
The viruses are entering very low on the food chain. Is that
At the
correct?
bottom, at the primary producer level. It depends on what the bacteria are, also, by the way.
So,
They could even
so
be-
phages could also be food.
That's right. That's right.
Sure.
And a lot of phages in the ocean. But
You bet.
oh wow, this is very cool. So the next time you drink some water, man.
Well, it's funny you should mention that because one of these little micro crustaceans is found in a lot in fresh water. And there is a rule among the ultra-Orthodox Jewish population that you're not allowed to consume crustaceans. And it was pointed out that the New York City drinking water contains these microcrustaceans at various times of the year, particularly in the spring and the fall. And so they had to give them special exemption that you could drink New York City water. They have a size limit now on the size of the crustacean that you're allowed to ingest. But you have, that actually came up as a religious thing rather than a biological thing.
So they say the growth efficiency was 17%. And this compares with other aquatic consumers. So in other words, viruses are good or a good
A
meal.
good energy source. It was a good energy
They're good
source.
eating.
Yeah, it was a good. They're good. Yeah, that's right. That's right.
Very interesting. I would really
Yeah.
like to see a concentration curve. How if you put more in, do they grow faster or more, whatever, or what's the limit? And different kinds of viruses. A lot of things to do here.
I was just thinking if you had, if you could distinguish the different kinds of viruses in an assay, you could even do a mixing experiment where you put in several at the same time and ask which titer decreases. If there's a
Right.
selective decrease in titer of one virus or another.
I suspect that. Well, I don't know that these organisms are capable of sorting them out that finely.
I wouldn't think
I mean,
so.
they're just kind of.
I would imagine that you wouldn't see it, that it would
No.
be non-selective. Still, this is where the mind goes.
Yes.
Now, we did a paper where the same group was suggesting that hosts can sense viruses. Does anyone remember that?
Don't remember that one.
It was the
Vaguely.
same.
Sounds a little familiar.
And my Google is not. My Google foo is not working here. Yeah, I'm pretty sure we did. So, what would be a good search term for that? It's probably chlorovirus, right? Chlorovirus and TWiV. Let's try that.
There's another virus. That
Oh, here it is. Multitudes contain me. Yes, the TWiV hosts present two potentially seminal papers. Hey, how about that? Two potential long-distance chemo attraction of a host by a chlorovirus. Okay.
Okay.
And
A
Kathy
chemo?
and Alan and Dixon and myself were all on that. A chlorovirus, the same one here. Chemo
Right.
attracts a host.
How? It doesn't secrete anything.
Tell them which TWiV to listen to.
It's
No, I listened to it. I was part of it, and I don't even remember.
well, it's
Well, that's why you need to listen again.
539. It was only March 2019, and this is a Journal of Virology paper. And what they found, we reported the novel concept. We demonstrate virus-packaged chemotactic modulation of prey behavior.
So that's my experiment.
Yes,
basically.
that's your experiment.
Yeah.
And what was the paramecium? So, they put a three-way thing on a Petri dish where the paramecium could go
sure.
one of three arms,
Right.
and they put virus in one, and it goes to the virus. Well, it's
Goes
a
to the virus.
beautiful
Yeah.
experiment.
But is that chemo
can smell
attraction, though? That the point is, what's the molecular signal?
I don't know how the virus is moving. It'd just be Brownian motion, right?
No, but it stays put, right?
Oh,
Yeah, the virus
is
is
virus
just what
can't-
the
sorry.
paramecium goes
Yeah,
to
it's
it.
the host
So
that's
what
moving,
is the molecular signaling?
The virus smells to the paramecium
Stop
like
it.
a
Well,
barbecue.
Stop it.
This is, I sat next to Dave Dunnigan at an ASV, and I asked him and he said, we're working on it. But meanwhile, they got waylaid by viruses being eaten, I guess. So.
Well, is there a way to know whether or not paramecium is sensing something soluble or particulate?
Sure, there is.
Sure.
Yeah,
They should have done that.
Put the appropriate filter in. They're working on it.
they're working.
They're
on
working
it.
on
They're working on
That
it.
was a while back. We should go back and ask.
They had to stop to feed the Halteria.
But then that, yeah.
Well, they may be listening, so
Yeah.
send us
With
an
any
email.
luck, they're
Here.
listening, and we'll get some mail.
Send us an email.
Because for me, chemo attraction has something to do with soluble compounds.
All right, let's do just one round of emails here. Dixon, why don't you start with the first one? Are you able to read email there?
I am not only able to, I will be happy to do so. Hi, Vincent and crew. It's from Anonymous, a good friend of mine. I've been enjoying your podcast for over 3 years now, so thanks for the excellent COVID coverage. Still haven't gotten the virus yet, somehow. 23-year-old male with 4 shots and regular N95 masking. That's probably why you haven't got it. I'd like to share a different virus-related story that happened to me just 2 days ago. Recently, a random dog that was being walked by a little girl touched my right jacket's sleeve for a second in Valencia, Spain. This prompted my rabies anxiety to go up by 1,000 points. I was going to eat when that just happened, so I cleaned my hands and sleeve with hand sanitizer, ate, and had a bit of a panic.
Got to eat, right?
That's right. You got to eat first and then you panic. Eat first, panic later. According to WHO guidelines, this is a category 1 exposure, touching or feeding animals. Licks on intact skin, contact of intact skin with secretions or excretions of rabid animal or person. So it's not regarded as exposure, therefore no PEP required. I won't be getting the shots after all. This doesn't quite put my mind at ease as health anxiety with what ifs kick in, but I'm staying rational. The guidelines are there for a reason. Thankfully, and surprising fact, Peninsular Spain has had no cases of dog-induced rabies since 1978, with only one imported dog rabies case in 45 years. This speaks greatly of the work that virologists, veterinarians, and the Spanish government have done over the last decades. I was also very sad to learn that the indication for potentially exposed dogs to rabies with no vaccine is mandatory euthanization. You loved your 1918 influenza episode with Jeffrey Taubenberger. You
Were
guys,
you doing TWiV back in 1918, Vincent?
Amazing. Yes.
I was.
Dixon was doing his big band jazz picks
That's right.
the
That's
first
right.
time around.
They didn't have big bands. They had just small bands back there. They hadn't
Right.
grown up yet. You guys are awesome. Keep the virology up.
I guess if Anonymous had known that there had been no rabies in Spain for all that time, they wouldn't have worried, right?
I wouldn't have worried.
Yeah,
at
this has everything to do with what the local regulations are, veterinary regulations for keeping animals.
A
'Cause,
symptomatic dog
and-
would have been easy to detect.
yeah, the very fact that this dog was being walked by a person,
Right. A cute little girl.
Yes.
is generally
No.
where you have rabies in dogs. It's where you have a lot of stray dogs and no control over the rabies,
Exactly.
Yes.
Right.
and then it's a real problem.
Oh, yeah.
But most
Yeah.
countries have it under control.
That's right.
Many countries have it under control.
Many
there's
developed countries generally have it
There's
under
an
control.
oral vaccine, right? In the form of a bait that they leave out.
For wild
them to
animals,
consume.
yeah.
Yeah.
All right. Let me take this next one because it's a little tricky. This is from Deborah, and it's a very long email about Deborah's experience in China, which we'll put online, but I can't read the whole thing. It would take a long time. Deborah writes, Dear TWiV experts, thank you so much for all the knowledge you have shared over the course of the COVID pandemic. When I, like so many others, found you, I've learned so much that is enlightening and recommended your podcast on COVID origins to all my Chinese politics specialist friends. I also have found Dr. Griffin's clinical updates personally invaluable, knowing what to ask for when. I'm writing you from Beijing, China, where I have been since the end of October. I've basically experienced it all. Zero COVID, the end of zero COVID, quarantine, the mass testing regime, getting COVID in China and recovering and now being out and about with a society, at least in Beijing, that is pretty much through the wave. I thought I'd share a little of my experience with you. Firstly, quarantine started with a quarantine hotel where one was literally confined to a room for 10 days. 3 times a day, someone would leave a tray of food outside my door, knock, and run away. I would see a fully PPE-clad worker hurrying down the hall as I retrieved my tray. Most days, a health worker, no idea what qualifications would come to give me a COVID test. At the airport, there had been both a nasal, very deep, and throat test, and that double procedure was repeated on day 7, but otherwise it was a throat swab. On day 7, they also swabbed various places in my room that I would have touched, doorknobs and the like, to test. I came back to my computer and found my trackpad wet. They had apparently swabbed it. All right, so this continues with all sorts of interesting anecdotes that you should go read in the letters section. But I'm not going to read it now. And I will go to the end where she says, I hope you find my tale somewhat interesting. I think it feels quite different on the ground. So this is someone in China, if you want to know what's going on. Deborah is an assistant professor of political science. Thank you, Deborah, for writing all of that.
Mm-hmm. It is interesting.
Cool. Yeah.
There's a lot of stuff in there.
Kathy, can you take the next one?
Rochelle writes, Dear Vincent, thank you and your amazing team of scientists and friends for providing me and thousands of other listeners with so much listening pleasure. Sometimes the information is over our heads, but we're always learning and appreciate all you have done for us these past years since COVID reached our shores. Today, you said that you believe the listeners learn from and appreciate the friendly banter, and you are correct. The back and forth between you, your co-hosts Brianne, Kathy, Dixon, Alan, and Rich create an amazing forum for learning complicated and vital concepts. I'm a recently retired pediatric nurse who just loves listening and learning about scientific research. Sidebar conversations make it all very entertaining and memorable. Having guests like Paul Offit and Carl Zimmer on the show reminds us that we have much to learn. Things are never stagnant or boring in science. I rarely watch, but thank goodness for your podcast, which can be accessed anywhere and everywhere. Finally, if you have fond memories of trips to the Horn and Hardart Automat in New York City, you can watch a wonderful documentary on HBO Max, maybe also YouTube, I'm not sure, created by Lisa Hurwitz about these amazing food emporiums that existed in a different lifetime in New York and Philadelphia that many of us of a certain age remember so well. Plus, the documentary is hosted by Mel Brooks, who sings and jokes in praise of eating creamed spinach in Manhattan so many years ago. Happy and healthy New Year to all. And thanks for all you do. I send a donation via credit card and purchased several items at vaccinated.us for holiday gifts.
Nice.
That's from Rochelle.
Thank you.
Thanks, Rochelle. Yeah.
Thank you, Rochelle.
Good.
Rich.
Jake writes, first of all, thank you for your podcast. Second, I was listening with interest to your episode TWiV 973, Crouching Virus Hidden Tigers with Felicia Goodrum. If I understand correctly, cytomegalovirus is widely dispersed throughout the body by way of infection, and a large part of immune function may be tamping down what could be a cytokine cascade or, at the very least, a serious immune reaction to the widespread infection by the virus. My question is this: could aging, or at least a portion of aging, actually be due to the body no longer being able to restrain the immune system from attacking CMV-infected body tissues? In other words, is aging largely or even primarily due to an is aging largely or even primarily simply an immunopathological disease that is triggered by an inadequate tamping down of a robust immune response to CMV-infected cells? Perhaps this could explain age-related symptoms like deteriorating joints, disintegration of cartilage, fibrotic changes in lungs, recession of the gums, or even some dementia. Perhaps these are manifestations of a slowly deteriorating check on the immune system in its quest to deal with CMV-infected cells, a quest that necessarily or eventually destroys or seriously harms the body because the CMV-infected cells will be injured and or destroyed by the immune system. Again, my thanks for TWiV are Jake. I'm going to let you guys take this. I was not on Felicia's episode. I have not listened to it. I do somewhere back in my library. I do know that or seem to recall that a significant fraction of your immune system is distracted by CMV.
That's right.
As time goes on.
Yes.
And it certainly makes sense to me that as you get older and your immune system loses some of its adequacy, that there could be problems. I question whether aging more generally is a result of this, but it could contribute to some of the problems. Comments?
Yeah, my understanding in talking when Felicia was on was yes, as you get older, what was it? She said something like a third of your immune system's responses become just CMV directed.
Yeah.
It just becomes this big focus. But my impression was that this is part of an overall decreasing of immune response, consistent with needing a higher dose flu vaccine and having a less reactive immune system. So this notion that it's an autoimmune condition, that it leads to an autoimmune response against your systems, seems a little out of sorts with that, but I don't know. So it's not totally off base. It just doesn't quite jive with what I've learned so far about the aging immune system.
I mean, also as you're aging, you're undergoing immunosenescence, right?
Yes.
So to postulate that an overactive immune response is causing aging doesn't make sense, right?
I'll let
Right.
you know.
Yeah, that's a better way of phrasing what I was
That's what you were saying? Okay.
trying to get at.
Okay.
It's it's more about the immune system is declining with age and not ramping up.
So, Rich, I hate to sound pedantic, but pedantic, but when you read the title of the episode, you missed it. It says crouching
did?
viruses, hidden triggers, not tigers.
Yes, crouching virus, hidden trigger.
Okay.
Right. Sorry.
Yeah.
That's
So that brings us to Alan, right? You haven't done one?
I have not
So
done
do
one
the
yet,
next
right.
two. They're short.
I'll do, it's a real short one and then a slightly longer one. Krister writes, "Hi, Vincent and the TWiV team. I just learned the plural of anecdote. It's obviously not data, but anecdata." Krister in Sweden. I've also heard it referred to as artisanal data.
I like that better.
That's
good. Handcrafted.
Catherine
handcrafted data.
writes, "Hi, TWiV team. I live in the Pacific Northwest. My best friend recently contracted COVID and called her doctor to ask about getting Paxlovid. She was offered the drug, but at the same time discouraged. She was told that if she wanted Pax, she should know that taking Paxlovid would make her more susceptible to it in the future. To me, this comment suggests that Paxlovid weakens the immune system. I had never heard this, and I think it is ridiculous and reckless. Please comment on this comment provided by a medical professional to a sick client. Thank you," from Catherine in Washington State. And yes, I think that was very poor practice by that doctor. I'm not aware of any evidence that Paxlovid is going to increase your susceptibility to COVID in the future. They were probably referring to what they think of as Paxlovid rebound, but as has been discussed extensively on the clinical updates that Vincent and Daniel do, that is not Paxlovid rebound, that is SARS-CoV-2 rebound. That's the virus coming back.
I don't understand why the doctor would do that, right? They're just misinterpreting.
I think probably they're misunderstanding the way the immune response works and thinking, well, if we tamp down the virus, then you won't have as strong an immune response, and then you'd be more susceptible. But that's not really how any of this works.
Doctors are people,
They're
not ferrets. They are not ferrets. They are people.
and
they are responsible.
Certainly had experience with doctors who state emphatically and with great authority stuff that I know is dead wrong.
Yeah. And they are responsible for keeping a lot of information in their heads at one time.
Yeah.
So, falling down on something as complicated and difficult as immunology, okay, that can happen. But yes, this is bad advice.
I recently visited a doctor, and we talked for 45 minutes about viruses. He was a surgeon, but he was really interested.
Gee, I wonder how the subject came up.
Well, I always bring it up. I like to talk about what I do, right? And then he brought me to the scheduler, and he said, this guy's a Columbia professor. He knows everything about viruses. Ask him whatever you know. And he was really embarrassing. Like, she's looking at him like, okay.
Right. No, I'm retired.
It's funny. All right, time for some picks.
Yep.
Dixon.
Okay. We continue with the Jazz Project. We're almost 3/4 of the way through this list. I hope you're not getting tired of it, because
No.
I enjoy revisiting all of this. Now that the instrument is now under consideration is the trombone. Now, I play the trombone, and I played it.
Do you play it well?
I play it well, not so well. I never really mastered the instrument to the point of being comfortable with it. And to say that I could just go up in front of people and play something. But I played for my own enjoyment, and I still do. And I still enjoy it, but nobody else does. So I play in a closed room with soundproof. No, that's not true.
You should bring it here and play here.
No, you wouldn't like that. you would. So here's my list of the trombone players that I really respect. I didn't start with Tommy Dorsey, because Tommy Dorsey was not really part of the jazz scene. He was part of the swing scene, but he played mostly bucolic, well-played music, but
Not jazz.
not what I would consider to be mainland jazz. So the first trombone player that really set the tone for all the rest of the trombone players was Curtis Fuller, which I don't list first, but I should have. Curtis Fuller was a magician when it came to getting complicated phrases out of his instrument, which are very difficult because there are no set positions on the slide. You have to guess where the notes are. And he had a wonderful ear, and he also played with lots of different groups. And you'll enjoy listening to his music. He's really a wonderful, inventive trombone player. This first two group, though, the Steve Toure and Conrad Hardwig, they are beyond amazing, just beyond amazing. Steve Toure has developed a series of performances in which he not only plays the trombone very, very well, of course, but he's got these conch shells fitted with a trombone mouthpiece. And he plays them, and they're all various sizes, and each one has a different tone range. And he does songs with the other trombone players that are there, only playing his shells. And he's got an album out called Sanctified Shells, and it's just absolutely wonderful. Conrad Herwig has a bit of a sense of humor, so the title of his signature album, A Jones for Bones Tones, sort of reflects his sense of humor, but he's an amazing, truly amazing trombone player. Curtis, like I said, Curtis Fuller is amazing. And then finally there was the duet of J.J. Johnson and Kay Winding. They played numerous great gigs together, and they produced many albums, and you will recognize them. And they played beautiful harmonies, and they were just absolutely the best of friends. So they knew each other, and they could anticipate when one would do something, the other would complement that. And they were a pleasure to listen to. And J.J. Johnson ended up playing with the Miles Davis Quintet for a while. Kay Winding went off on his own and played. In fact, he played the graduation of the Fairleigh Dickinson class that I graduated in. So the after party for that had the Kay Winding Quintet, and three of those players were trombone players. So I really I respect the instrument and respect the people who actually try to take it up, because it's not an easy instrument to learn, and to stay with it long enough to say that I now know what I'm doing requires lots of patience and lots of talent, and neither one of which I've got.
Nice. Trombone's a good instrument.
I love it. It's got a great tone, and it's wonderful to hear, especially when you play Dixieland music, and that I'm more comfortable with as a group, because there's more ad-libbing and there's less written music and more from the heart music, and so I like to listen to the trombone.
I've heard you play.
Yeah, you have. I'm okay.
Dixon, my cousin is a trombone player. He
you
was
mentioned
formerly in the Coast Guard band, and he talks a lot about playing loud. And so one time he said, "Guess what we do when the dynamic marking is 4 Fs?" And the answer is, we play as loud as physically possible, like a lot of the other times.
Yeah.
It's funny, but he must be very good, because there are lots of musicians in the Navy that don't make that band.
This is
Part of
Coast
the-
Guard, but yeah.
Coast
Yeah.
Guard, well, that's true also for
Yeah,
all
I
of
mean,
armed services.
yeah, auditioned specifically for that band. But and then Richard Strauss, the conductor, never look at the trombones. It only encourages them.
Right. I think classical music is a little bit more.
that's
But
good.
you know,
There's
when you
there's
listen
a
to
list
Bolero,
of jokes.
the music by, come on, Kathy, help me out here. Who wrote Bolero?
You scared it out of me. Yeah.
So that person, there's a trombone piece in there that's absolutely melodic and wonderful, and just Ravel wrote
Right.
Ravel.
Bolero,
Thank you.
and
Yeah.
just the trombone piece in that is just worth a listen. It's just absolutely wonderful.
So
Do you know how many trombone players it takes to change a light bulb?
Too many.
Only one, but he'll do it too loud.
That's unfair.
I just searched for trombones in rock music.
Well, Chicago had a great trombone player.
Chicago is
Oh, yeah.
Blood, Sweat, and Tears.
Really fabulous trombone. And by the way, the connection between him and myself was that I eventually learned through reading an article about him that he likes to tie flies just to pass the time of day.
Mhm.
He doesn't fish. He just likes to tie flies. So,
A friend of mine in college was a trombone player with a rock band.
yeah.
I think it's an
Yeah.
interesting combination, right?
The Almighty
It is.
Senators.
A lot
if
of
anybody wants to check them out.
Latin music, a lot of
Yeah.
salsa has great trombone players, really fabulous. So this is a partial list, obviously. These are my favorites, but there's so many wonderful trombone players out there to listen to. Steve Davis is another one. Steve Davis, I was on an airplane flight from Newark to Pittsburgh, and I remember this distinctly. I see this guy get on the airplane. He's got his trombone with him, puts it on the overhead. Anybody that puts their trombone on the overhead probably is a professional trombone player, right? So he gets to go off, and I'm right behind him. And I said, looks to me like you're a professional trombone player. He says, yeah. I said, may I ask who you are? And he said, I'm Steve Davis. I said, you're Steve Davis? You know what he was doing? He was going to a recording studio in Pittsburgh to work with Conrad Herwig on that, Jones for Bones Tones.
Cool.
That was the call date for that.
Very cool. How did he fit it in the overhead? It's pretty long.
No, but it doesn't take up too much room. And he said, you've actually heard of me?
Yeah.
I said, I think you're fabulous. He says, oh, gee, thanks. No, he was such appreciative person and very nice.
Sometimes famous people can be appreciative.
Yeah, well, I don't think he's famous, but I think he's a wonderful trombone player.
He's not famous? Not at all?
I don't think a lot of people know who Steve Davis is.
Among jazz aficionados, probably.
Oh, yeah, absolutely.
Yeah.
No, there's no question about that. And I think, blocking on his name, the guitar player that used to play on The Tonight Show has a brother, Kevin Eubanks. And Kevin Eubanks is a trombone player of the first order and an amazing. In fact, I'll add him to my list right here. Kevin Eubanks. I'll update this and give that to you.
Yeah, so Bert Semler told me a story, Kathy. He was sitting in a restaurant, and Reggie Jackson was at the next table with his daughter, the baseball player. And so Bert said, should I say hello? So he leaned over and said, I just want to say I really enjoyed when you played for the... And he loved it. He was grateful. He said, oh, thank you so much.
That's right. That's right. Do you want... I can do a follow-up on that. Michael Katz was the chairman of pediatrics at Columbia for many years, and Reggie Jackson walked into Baby's Hospital, and he wanted to just visit the sick kids and say hello to them. And there was a guard there, and the guard said, can I help you? And the guy says, yeah, I just want to go see some sick kids and just make them feel a little bit better. And he says, well, who are you? He said, I'm Reggie Jackson. He says, I'll be right back. So he goes and he calls up the chairman of that department. And Michael Katz comes out and he looks at the guy and he says, you can't come in here. These people are sick. And he kicked him out of the hospital. And it was Reggie Jackson.
But
if he knew who Reggie Jackson was, he would have let him go ahead.
Sick people have visitors.
Of course they do, but he thought he was just some guy that walked in off the street that wanted to, they get a lot of that in New York City also.
Who was the actor who was short and he danced a lot?
There are many.
In movies.
Mickey Rooney.
Mickey Rooney, right. So I'm sitting at having breakfast before going to Amgen years ago. And Mickey Rooney I didn't have the guts to say
Oh, you should have done it.
I got in an elevator once, and Bill Nye, the science guy, was there.
Yeah.
And I didn't say anything to him.
I was eating in a... we can go on like this forever, but
I'm sorry.
We could go on like this for a long time, yes.
We were in a Cafe Luxembourg, which is on 70th and Broadway. I'm having sandwiches with somebody else, and I look across the way, and there is Mr. Spock, right?
Oh, Leonard Nimoy. Yeah.
Leonard Nimoy was there with his granddaughter and family celebrating his granddaughter's birthday. And
Did you do this?
I not only did, he did that first. I had to go over there, and I said, look, I enjoy all of your portrayals. They hit at the ethics of being human. And you've taught us great lessons through your character. And he just melted. He just melted. Turns out he was suffering from COPD, from heavy smoking, and 2 months later he died. But as I walked out, he looked back at me and he went like this.
That's nice.
And I was so touched by that.
If you're civil, people will be nice to you, right?
Absolutely.
All right. Kathy, what do you have for us?
I have something that we don't often pick. This would be like a mini snippet, but I picked a paper that is about measles and subacute sclerosing panencephalitis.
Yes.
We have a feature in our class called Virus in the News, where for the first 5 minutes we talk about something. And famously, in early January 2020, we talked about this mysterious pneumonia in China. And So this one is a paper that was suggested by one of the students, so that makes it extra special. She suggested a link from neurosciencenews.com, which is a nice, easy lay version of it, and then the primary research article from Science Advances. And so it's explaining something about how SSPE, which is one of the sequelae of measles virus infection, can develop. And they have a nice figure in the lay version that kind of explains it. But basically, they found that the fusion protein of measles, which is the F protein, can have mutations that increase infection activity and other mutations that decrease it. And it's thought that these mutations that increase the activity can at first be overcome by the normal F protein being there. Oh, and this is because they think that there are co-infections going on. And so that can be overcome by the normal F protein. But then eventually there's so many mutations that accumulate that then the F protein, the mutant one, has picked up so many mutations that it can lead to the fusion that causes SSPE. But the other thing that's cool is that they find in this that there are some mutants that can't fuse. And so then they have this model whereby they think that the mutations or the F mutations decrease the fusion activity. And so the ones that decrease the fusion activity are somehow helped by the normal wild type protein, and that leads to SSPE. So they sort of have two mechanisms by which you can get to SSPE. And so I have the primary link in there too, which is in Science Advances. And then I sent the slide picture that I modified a lot to Vincent and Rich earlier in this week. But I just thought it was a cool story that is trying to get at the mechanism for how this virus, which the sequelae is that, it comes up 6 or 8 years after the initial infection. And it's 100% lethal, and there's nothing that can be done for it when that happens.
Right.
But it's thought that it's the virus infection initially that just persists, and then somehow these mutations can accumulate, and that's how it
Was this one of the first viruses they called a slow virus?
I'm not sure that they technically call it a slow virus, but I think of that mostly as being lentiviruses.
Because Michael Katz, the man I just mentioned, who's exiting Reggie Jackson from Baby's Hospital, worked in Philadelphia at the Wistar Institute and was partially responsible for rescuing this virus from a brain of a woman who had died from SSPE.
They say on the web, it says SSPE is an example of a slow virus infection.
Okay.
Right, so that's where I learned it
Okay.
because he was my chairman for a while. So I got a lot of insight into how he conducted his research.
It's very cool. I like
Yeah.
this. Yeah.
Yeah.
This,
Neat.
This is worthy of inclusion in a lecture.
Indeed.
Very cool. Because they usually say, we don't know what's going on. So
Yeah.
get some ideas. Rich, what do you have for us?
All right, before I do my pick, I want to do a real quick sidebar TWiV experiment. That phrase at the top of the show, CCBNA, chit chat but no action,
Yeah.
that came from a college roommate named Dan Hammer, who I think spent part of his career, on Dianne Feinstein's staff.
Oh, wow.
Okay, he was into politics. Okay, and I have lost touch with Dan. And I miss him even today and quote him with that phrase all the time.
Yeah.
TWiV has enough of a reach so that there's got to be few enough layers of separation between me and between TWiV and Dan that somebody could get back to me.
Yeah, could be.
There you go. We'll see if that works.
All right.
Okay. My pick is what has been echoing around the media recently, which is the announcement by Colossal Biosciences that they were going to de-extinct the dodo.
Right.
This is their third announced de-extinction The first being the woolly mammoth, the second being, I believe, the Tasmanian
tiger.
and now the dodo. And the reason I picked this is because my wife was reading the article and she noticed something about the techniques, which is that basically the technique that they propose is to pick the closest living relative that they can find and take what they know from dodo sequence and edit that into that close relative and come up with something approximating a dodo. Okay. And she wanted to know why that, instead of just slamming the whole dodo genome into some appropriate egg. She knows enough biology to be familiar with Gurdon's experiments with frog intestine nuclei and stuff. And so that got me going, and I had to look all this stuff up. So the pick has kind of sort of three parts. First, the title. The first, it links to an article in The Hill, but the title of the link I had to retain came from our paper because it's got a marvelous pun in it, which is "Firm hatches plan to bring back the dodo."
Yuck, yuck, yuck.
And then there's a lay article, which I couldn't link to our paper somehow, but there's a lay article in The Hill that describes the whole thing. But that got me looking up Colossal Biosciences because I wanted to know who these guys were. And lo and behold, it turns out that they are a company that was launched by a guy who is described as a serial entrepreneur, Ben Lamb, and none other than George Church at Harvard University, who it turns out, if you look him up, has launched in his lifetime something like 50 companies and holds the Guinness Book of World Records for launching something like 18 in a single year. I encourage you to look through Colossal Biosciences and come to your own conclusion.
I've seen the movie, Rich. I've seen the movie.
One thing that really struck me is this quote under the company info by a person named Letitia Garriot-Dechaillou, who is the founder and CEO of Global Space Ventures. Quote, "In 2014, I asked my friend Elon Musk who he knew who was poised to make profound changes in the field of genomics, and he said, George Church." So that means that George Church is the genomics equivalent of Elon Musk. So take that for what you
Take that
want.
how you like.
So, Rich, how many companies that he established to begin with are still in existence? That's what I want
I
to know.
don't have that number.
I bet you
don't
A
have
somebody
that
lot
number.
does.
fewer than he's
Wall
established.
Street
one
has that
thing-
number.
One thing for sure is that Colossal Biosciences are really good at PR
Oh, yeah.
because they've raised a lot of
No,
money.
you're right. You're right.
All right.
And the web page is
and
gorgeous.
The website is amazing. It's really good.
I'm sure.
And they have a whole section on the dodo that
I'm
gives
sure.
all the background
Yeah.
on the dodo.
There's no trouble
and
finding
and it's
them.
beautiful.
They should try some living dodos, though. There are plenty of them out there.
I thought it was
Right.
extinct.
No,
Rich,
no.
what-
Other dodos.
Oh, oh.
Is it proprietary, or have they indicated what genome they're going to start with that's closest?
Yeah,
Right.
some pigeon, a particular pigeon.
Really?
Ah, okay.
Okay.
Dodos are in the
close.
dove family.
So they're using the same strategy in all cases, where they start with the genome of a close relative.
Right.
In the case of the mammoth, it's an elephant of some sort, and in the case of the Tasmanian tiger, it's another tiger.
Not a dog? Not a dog?
No, I don't believe so. I think it's another tiger. I could look it up and verify it, but I'm not going to do that right now. At any rate, so and then, the idea is to, from whatever information they can get out of the extinct genome that they're trying to clone, is to identify. This is one of the many tricky parts. Identify genes that they believe will confer the appropriate character to for the extinct creature,
Right.
and then genetically get them into the host or scaffold genome and reactivate
So,
that.
Rich, it's quite amazing. Oh, I'm sorry.
I presume, among other things, that these, trying to figure this out rationally, that what I wound up telling my wife is that, the genome sequences we have for extinct creatures are a gazillion fragments
Correct.
that we have put together virtually on a computer, and it's probably not perfect. It's probably far from perfect. It's probably got gaps and mistakes and whatnot in it. And if you think about it, although theoretically it would be possible, assembling that into real chromosomes that you could stick into a host egg of some sort to launch an organism, I guess is judged to be technically less feasible than doing all of this engineering to use a sort of a host genome modified to de-extinct the organism. That, of course, doesn't even address the question of why you want to do this.
Right.
Okay? Or, in fact, I think that there are very interesting ethical questions associated with this.
Yeah.
Okay? So, at any rate, that was my, I had to do this because I just got off. Okay?
Is,
I couldn't stop myself.
What do you?
They have a whole hype ecology around what they're going to do with this and reconstructing the ecosystems in which these animals lived. That I can understand why somebody like Elon Musk would be into this,
It's a
because
bit
they run on the
grandiose,
same
wouldn't you say?
same kinds of
Do you
grandiose
think,
frequencies.
Rich?
Yeah, go ahead.
You think this is going to work, Rich?
No. But, I'm a pretty poor judge of this kind of stuff. Because, if well, I don't know. I don't know what I would have said about SpaceX. I might have said SpaceX might work. But there's a lot of things that I've said aren't going to work that have worked. So I don't know. But it sounded pretty far out to me.
This thing, by the way, the thylacines were marsupials.
Right. But marsupial,
and they are carnivores. Their most closely related living relatives are things like Tasmanian devils
dog-like creatures.
and quolls.
Right.
So I think that's what the source genome is going to be.
Okay.
Yeah, that's probably right. So, this is, I don't know. I would say this is a fool's pursuit. But today I addressed an undergraduate class with Darwin's finches and gave them the genetics of that. And I said, I know none of you are interested in genetics, but you might be interested in learning about the fact that the beak of the finches is under the control of a single gene. And so what they've done is that they've transfected chickens with each of Darwin's finches and gotten a chicken beak to look like the Darwin's finch beak.
Huh.
So there may be some stuff to this, like the
you're
beak
going to learn
of
something.
a dodo.
Sure.
It may come all out in a single gene. Who knows?
So, do they plan to bring these birds back to Mauritius? Should they work?
I haven't gotten that far into it. Well, as Alan says, this is associated with bringing them into appropriate ecosystems. But they don't expect these to be, exact replicates. They even have little things about how they're going to be things that, would be adaptable to current environments, okay, relative to, say, the environment they came from. How you judge that, I don't know.
Yeah.
So, if we could do it, should we bring dinosaurs back?
I think that would be a big waste of effort.
Waste of
What
effort?
you would get
Birds.
back would not be the same thing that was gone.
But
And these are not going to be dodos, and these are not going to be mammoths, and these are not going to be thylacines. They're going to be some product of genetic engineering that resembles it superficially, and then what do you
If
do?
at all.
But if they can make T-Rexes with little arms,
Little arms, yeah, that could be cute.
yeah, well,
If they could make the whole T-Rex small, that might be
but then they're going to get out and destroy things.
Yeah.
You know?
right.
yeah.
Maybe.
we all saw the
That's a
movie.
gain of function
But
that I
you
don't
know,
want to see.
Yes.
Oh, yeah, that's the other thing I thought. This is gain of function on
This
steroids.
is gain of function. Yes.
I think if you do it with wheat, it's a grain of function.
Look, I think it's interesting scientifically to try it, but I think dinosaurs would be fascinating too, because then you could, I know they're not going to be dinosaurs, but you could study some aspects of them.
They're missing.
I would like to know what color they are. what their skin looks like.
Whatever color you make them.
Yeah, it's a thing. You can
True.
make them without. Very interesting. All right, Alan, what do you have for us?
Okay, I'm taking a brief departure from my my video game picks because today this was something that I thought would be topical for people who are dealing with these weather conditions. This is the live Mount Washington, New Hampshire observation camera.
Oh, boy.
And you can go ahead and play this, and you'll see very little besides a little bit of a ledge and a whole lot of snow, and you can see the camera shaking.
Bet.
Yeah.
Mount Washington, New Hampshire, until sometime in the mid-1990s, they held the world record for the highest recorded wind speed on Earth, which was something like 231 miles per hour. And then somebody finally recorded a wind speed greater than that, like in a typhoon somewhere. But this is a place that is noted for having some of the world's worst weather because it's not that it's such a tall mountain. It's only about 6,000 feet, but it's just not. It's surrounded by nothing
That's right.
nearly
That's
as
exactly
tall.
right.
So there's nothing stopping this wind that's just blowing through the atmosphere and screaming across this mountain. And they're expecting the wind chill there to be around minus 100 later today.
This is what state? I'm sorry.
New Hampshire.
New Hampshire.
New Hampshire.
And you can buy a bumper
You can-
sticker
you
that
can
says
climb it.
"My
Yeah,
cars."
I've been up there.
Yeah.
Yeah.
Cool.
In real time, we're watching people.
Yeah,
join it.
we're watching the Mount Washington camera in real time, at least until it blows away. I don't know how many cameras they go through in a year, but by the way, in the course of this recording, my temperature, as reported at Westover Air Base just up the road, is now 5 degrees Fahrenheit, minus 15 C, and wind is gusting to 38 miles per hour.
The worst is yet to come, my boy.
So there you go.
My pick is a website I stumbled across and seems to be pretty good. The parent website is immunize.org. They have a lot of information there, but the page I found is all about vaccines. Experts answer questions about vaccines. So they have a lot of vaccines listed. You could pick one. Let's see what they have to say for polio. What's the current state of polio in the world? What is the routine schedule for IPV for children? Should adults get vaccinated? Yeah, a lot of interesting questions. If you do influenza vaccine, it's equally comprehensive. So I think it's legitimate and looks good. Here's one: vaccine recommendations for different places. They have links to package inserts, ACIP recommendations. So it seems to be maintained by 3 MDs. And I found it useful. So you might check it out.
This is this is a good organization. I've actually worked with them. I covered some conferences that they co-sponsored. They do the what is it now called? It's like it used to be called the Flu Summit, and now it's the National Adult Vaccination Summit they have every year, which all the big entities in vaccinology get together in Atlanta every year. And immunize.org is run by LJ Tan, who was our guest on TWiV 103.
Yeah, he's
Way back
he
when.
has expanded in prominence since
Yes.
that episode.
He has
You
definitely
got the
expanded
TWiV
in
bump. You got the TWiV
prominence
bump.
and
Come on.
right.
Well, he got the COVID
Came much
bump.
later.
I think he got the COVID bump.
Yeah. So he's been very much at the forefront of vaccines for his whole career.
Yeah. We have a listener.
But-
Yes, go ahead.
I just looked up the weather on Mount Washington right now. Minus 42 Fahrenheit, which interestingly is minus 41 C, and 90-mile-an-hour winds.
90-mile-an-hour
Just
wind.
90 miles.
That's a light breeze for Mount Washington.
Exactly. Let's go hang
Sorry,
gliding. Come
Vincent,
on.
go for it.
We have a listener pick from Roberto. Dear TWiV team, first, like many other listeners, I want to thank you for the invaluable work you do in science communication. I've been an avid listener since mid-2018 when a colleague of mine who is a chemical engineer recommended your podcast. Needless to say, during the pandemic, your program became an essential listen. because not only did it keep me sane and well-informed so that my wife and I could make informed and intelligent decisions about our health risks, but it also turned me into a sort of guide for some friends and family who were inundated with alarmist news and misinformation. I was even able to manage my elderly mother's pandemic strategy from thousands of miles away, all thanks to your advice and clear statement of the facts as you knew them at the time. I want to suggest a listener pick, which is right up TWiV's alley. My colleague Andrew Wehrman just published The Contagion of Liberty, The Politics of Smallpox in the American Revolution with Johns Hopkins University Press. The book explores the emergence of the scientific discovery of inoculation for smallpox in the West and its confluence with growing political concerns in the early years of the U.S. Republic. It explores the details of how the origins of inoculation were whitewashed so that it would be more acceptable to white colonists. It also documents how prominent American figures such as Benjamin Franklin became some of the strongest advocates for inoculation, thereby making it an American discovery. It is not a dry academic book, but rather an engrossing read that sheds new light into the history of public health politics in the U.S. Really is worth reading. It sounds great.
Yeah.
Mm-hmm.
Again, my sincerest thanks for all you guys do. By now, I am so used to your voices and quirks that I feel like I am listening to a group of trustworthy friends. Keep up the good work. Roberto is an associate professor of history, world languages, and cultures at Central Michigan University. Cool.
Cool.
Excellent.
Very cool.
Right.
So
Maybe we can repurpose the recruiting poster that shows Uncle Sam rolling up their sleeves.
that's right.
Yes.
In this case, it's to get vaccinated.
So today I had some movers move the remainder of my office things here to the incubator from Colombia. And the guy who did it, he came in and he looked at the Ebola virus on the wall. And he said, "What are you doing here?" And so I explained to him, we made podcasts to explain science. He goes, "Are you like Dr. Phil?"
Dr. Phil.
I said, no, I'm not a medical doctor, but we try and
Neither
explain
is he.
things. He said, "Oh, well, maybe you would know this. These variants, are they going to keep coming?" I said, "Yeah, probably." And I gave him my
Yeah.
card. I said, "If you have any questions, and I'll ask Daniel Griffin,
It's funny.
because he has an old mom up in the Bronx." I said, "Oh, yeah, if you have issues, I'll talk to Daniel." It's cool. You got
There's
to spread the information.
There's a bit in this letter I like. I was even able to manage my elderly mother's pandemic strategy from thousands of miles away, all thanks to your advice and clear statement of the facts as you knew them at the time.
Yes.
That means he got the message.
Yep.
He's
Yes.
been listening. This is science.
Yeah. Yeah.
That's right.
It moves.
Did I skip that?
No,
No, you
you
read
read
it.
that.
Oh,
You read it.
Rich was just rereading it for emphasis.
Yeah.
Actually, I didn't remember reading that. It's good, though. It's very
Yeah.
good. All right. TWiV 981. Show notes are at microbe.tv/TWiV. You can send questions, comments, listener picks to TWiV at microbe.tv. And if you enjoy our work, we would love for you to support us financially. We are a nonprofit organization, and you know what that means. So go to microbe.tv/contribute. Unfortunately, Amazon Smile is stopping. It's too bad. We've got almost $1,000 since November from Amazon Smile from all the people who are doing it. It's too bad. Mr. Bezos, come on. Right, Dixon?
Right.
Well, speaking of Dixon de Pommier is at trichinella.org and thelivingriver.org. Thank you, Dixon.
You're welcome.
You like
This is-
coming here?
I love coming to the incubator. This is a lot of
So we're sitting at this table, and Dixon before says, "This is too wobbly. You have to tighten it."
and he did.
So I got underneath with an Allen wrench, and it made a
Now
big
you can't
difference.
move it. It's like
Thank
a
you.
rock.
It's very nice. Thank you. Dixon is a carpenter at heart.
I was a carpenter professionally during my college days. I made kitchen cabinets in the summertime.
Wonderful. You
Yeah,
should do it.
it's
You
fun.
could make a lot of money.
Not a lot of money. No, you can't make a lot of money, but you can make some money.
All right. All right. All right. Kathy Spindler is at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at alandove.com and theturbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.